Hello everyone, welcome back to KaushalBioPrep. In today's video lecture, we are going to talk about another topic, that is the modern methods of breeding for sheep and cattle. So first of all, why we do livestock breeding? So the aim is to improve traits like milk yield in case of cattle, meat quality, wool production, growth rate, fertility, and disease resistance. So that means whatever the production is, we want to increase that, right? Next, the traditional breeding mainly relies on the natural mating, which we have already discussed in the previous lecture. Selection based phenotypical traits we choose, but this is slow and it is also less precise. So there are some modern methods which are developed using biotechnology, genetics, and reproductive technologies, and they accelerate and basically refine the improvement programs. So modern major methods for breeding for sheep and cattle are here. These methods include, first of all, artificial insemination. Then we have the second one, that is called the embryo transfer technology. Then the next one is the multiple ovulation and embryo transfer. Then there is one more, that is the in vitro fertilization. It has multiple variants in vitro fertilization. Then there is marker assisted selection, that is the MAS technology, DNA based technology basically. Then there is genomic selection. And then finally there is cloning. And coming to the next one, that is the Transgenics, which is basically developing right now. It is an emerging technology. And then there is controlled mating system, which we have already discussed in the last lecture, right? So these are some major technologies which have been used for livestock breeding. So we will be discussing each one of these one by one. So let's start with the first one, that is the artificial insemination. So this is in this basically we collect the semen from the genetically superior male species and artificially introduce it into the reproductive tract of the female at the optimal time of ovulation. This is very important. When female is ovulating, only then this is going to be effective, right? So ovulation time is recorded. So at the optimal time of ovulation, the semen from a genetically superior male is artificially introduced into the reproductive tract of the female. So the steps are: number one, semen collection from the genetically superior male. Then there is semen evaluation. You evaluate the semen for their characters. Then there is processing and storage of the semen. And then heat detection in female, that will basically give you the information about the ovulation. And then there is insemination. Okay, in some in some females or some species, we call it heat period also. The ovulation period is also called as heat period. That means the female is reproductively active, right? So application of this particular technique is this is widely used in cattle breeding. We use it and then it is also increasing in the sheep with the laparoscopic AI. This is something which we are developing right now, so we will not talk about this. But in cattle breeding, this is commonly used, right? And it allows us of proven siren across the large herd of or the flock. So we basically use those males which have been proven to have the upgraded characters. Now advantage is that this is rapid dissemination of elite characters, so quickly you can decide. And then there is a reduced need to keep dangerous breeding male on site. You don't need that. Then there is help control the sexual transmitted diseases, because there is no sexual contact, right? Because we are doing the insemination artificially. It is also cost effective when you are comparing it to keeping the multiple breeding bulls and rams, because if you do the natural breeding methods, you need to have all the bulls and rams around, right? So it is cost effective. Then limitations are there. It requires trained technician, of course. A normal individual which with no prior knowledge of artificial insemination cannot do it. And the proper heat detection is very important. If the ovulation period is not going on, you are basically going to waste those semen. And semen handling is very important, and storage is also very important, and that requires the infrastructure formation, right? Next one is the embryo transfer technology, that is called ETT. In this technology, basically fertilized embryos from a superior female are flushed out. So you collect the fertilized embryo from the superior female and they are transferred to the surrogate mothers, right? So embryo are transferred, right, to another mother. So the process is super ovulation. This is done with the help of the hormone therapy to the female. Then there is insemination. This is again, you can have the required semen, the desired one, with the required characters. Then embryo are taken out from the female, this superior female. Then the quality will be checked and it is transferred to the synchronized recipient. The recipient needs to be in the synchrony with the cycle of this female also, right? Then the application of this procedure is, it is the multiplication of the elite females, particularly valuable for breeding with small population, okay, because it is a female based concept. So you will be multiplying the elite females, right? Advantages are, one elite cow or ewe can produce multiple offspring per year, because the reproduction procedure is long and it takes time, and along with that it also deteriorates the body of the female, right? During the parturition process, it weakens the body of the female. So if you are not using the female for that process, you are just collecting the ovums, or in this case the embryos from the female. So you are not basically letting the female have the whole process of the reproduction, and that in that case, if this a particular elite cow or ewe can produce multiple offspring per year, right? And genetic improvement is also rapid in this case, and there is preservation of the valuable genetics. What are the limitations are, this process is costly and technically demanding also, and it also requires the synchronization of the donor and the recipient cycle. The next one is the multiple ovulation and embryo transfer. In this particular thing, there is advanced ETT. That means this is embryo transfer technology, but this is advanced form, and it can combine the hormone induced multiple ovulation with embryo transfer. Now this is the advanced version of the embryo transfer technology. What happens basically is that breeding scheme, the use of the breeding scheme in this particular technology, I'll come back to the picture, is that it is the foundation of the nucleus breeding scheme, where a nucleus herd or flock supplies improved genetics to multiple herd. That means from one particular herd you are supplying the genetics to different herds, right? Now but what basically you do in this, so this is the superior bull. Semens are collected in the cryo container because the temperature needs to be maintained. Now this is the sperm capacitation, which can be done in the over the dish, right? You improve the quality of the sperm, that can be done, right? And then there is mating. So this is the super ovulated genetically superior buffalo. So what happens in this case is that the embryos are collected in this case, but you have already given the hormonal therapy to this particular female, so that the number of the ovums increase, right? So if there are multiple ovums, so there will be multiple zygotes. So these zygotes are collected, okay, and then they are cryopreserved again, right? Again, from these zygotes, one more thing can be done. From these zygotes you can collect different cells, right? Basically the ICM, the inner cell mass cells from the blastocyst, and you can also have the 2 embryos or 3 embryos as per the basically the development of the number of the ovums in the female. So these zygotes are kept in the cryo container, and then these can be used further in the surrogate mother. Like in this case, you see the embryos are transferred to the estrous synchronized surrogate mother. This is very important. Both needs to be in synchrony of their cycle, and then you will confirm the establishment of the pregnancy, and this surrogate will give birth to the new elite animals, right? So basically in this case you are using the hormones to increase the number of the ovums. In this case, in the embryo transfer technology, sometimes you don't use hormones to increase the number of the ovums, right? So simply you just take the fertilized embryo from the elite female and then transfer it to the surrogate mother. That is the only difference in both these cases. And right now people are also using AI for this procedure, right? The sperm capacitation and other things. Now, what is the benefit of all this? So benefits include the enhanced genetic gain by shortening the generation intervals, that you understand, and combine well with the open nucleus breeding system that I basically explained over here. Constraints are this is a very expensive procedure. The hormones are very expensive, and variable response to superovulation has been seen. That means that a single female will respond to hormones in different way. You cannot assume that all females will respond in the similar way, because every body is different, right? So that is the constraint of this study. Now the next method is in vitro fertilization. So fertilization occurs outside the animal's bodies. So oocytes are collected. In this case, now you are collecting the oocytes out of the body. They are collected from the donor, fertilized in the lab, and consulting embryos are transferred, right? So again, you are not taking out the embryos, you are taking out the oocytes, and fertilization is occurring in the lab. Now, what is the application? Application is the rescue of the genetic from valuable females with fertility issues. Sometimes the female cannot contain the pregnancy due to some constraints, or fertilization cannot be happen in the body of the female. The female have a fertility issue, so you collect the ovums, right, and preserve the rare breeds. Advantage is rare year round embryo production, because embryos are produced every estrous cycle. So you can basically ovum are produced every estrous cycle, sorry about that. So you can have the embryo production year round, right? And works when natural conception is not possible. This is important. When you cannot make sure that mating is there, even after mating there is no conception, so this procedure is done, right? It is also in vitro fertilization is also very very common in humans also, right? Test tube babies, if you are familiar with it. The limitations, it is high cost, of course. This requires a lot of money, and it requires advanced laboratory facilities also, because they are taking out the germ cells, so they needs to be maintained in a particular environment. Next one is the marker assisted selection, that is MAS. This basically uses the DNA markers and link to the desired trait to select breeding stock. So you are using what? DNA markers which are linked to your desired trait. So example markers are a milk protein gene in cattle, and you have the scrapie resistance in sheep. You have resistance to internal parasites in both cattle and sheep. So these genes are basically selected, right? These DNA markers are selected, which are linked to the desirable trait. Now the advantage of this particular thing is the greater accuracy than selection based only on the phenotype, because you are talking now on the genotype. So genotype based selection is actually more advanced than phenotype based selection. Early selection possible before the traits expressed, because you know the particular expression of the trait. Then use full for trait with low heritability. Sometimes a trait is very very important, very useful, but it is not inheritable in the same manner. The percentage of inheritance is different, so you can basically just select that particular trait from the DNA marker. And constraints are, they require molecular biology experts, and it is initial cost for genotypic is there. So you have to invest in genotypic. You first need to know the DNA markers, only then you can select, right? Then there is genomic selection. So in this case, you predict an animal's genetic merit using something called genomic wide SNP. SNP is single nucleotide polymorphism data. Now what is SNP? Basically, in the whole DNA, in a single, these are your nucleotides. In a single nucleotide there is a change, and this change is going to be different for each individual, right? So this is like a marker for that particular individual. The SNP varies, right? That is what is called the polymorphic thing. The single nucleotides is variable in different organism, so you can use it. Okay, so you can use it in prediction of the genetic merit of an animal. So in this case, the process is the first of all the reference population with phenotypes and genotypes. You select the population, then there is statistical models to analyze, then finally there is something that is called the genomic breeding value. This is important, this you need to basically remember. So what you do, you take a population, statistically analyze the population, then comes out with the genomic breeding value based on the pedigree analysis, that is the history of the population, right, over the generation. Then genomics and performance, all these are collectively giving you one thing, that is the EBV, that is the genomic breeding value, right? This breeding value is analyzed, and based on that you select your organism for further going on. Application in dairy cattle, it is used extensively for bull selection. जिसका this value is more, that bull will be select, right? Then sheep, may it is still working, we were still working on this is still emerging, you are not using it that commonly that we are using in cattle. Advantage is that it increases the selection accuracy also, because now you have a merit on which you are basically selecting your organism. Then there is reduced generation intervals and accelerates the genetic gain for complex traits. But there are some limitations, that it requires a large reference population you need, and that population needs to be large, and again you require the infrastructure and data analysis needs, right? Finally, there is cloning. So in cloning, you produce a genetically identical animal from the somatic cells. So there are two types of cell in the body. First is a somatic cell, and then there is a germ cell. The ovum and sperm cells are the germ cells, right, which carry the information to the next generation. Somatic cell usually do not go to the next generation, but you can use these cells to produce a genetically identical animal. For example, we have already done this with a very famous example, that is the Dolly the sheep in 1996, the first mammal clone from the adult cell. So in this case, what the scientists actually did that, so there is this adult fin dorset ewe, from where the nucleus, it is a nucleus donor. So donor cells is removed from the mammary glands of this particular sheep, and donor cells, which is the normal growth cycle, the donor cells starved, right, in the low nutrient culture medium. From there, the donor cell basically arrested growth, because they don't have the nutrients, so they went into the arrest, cell arrest जिसको आप कहते हो. Finally, these cells are basically fused with another cell, which are basically coming from the another sheep. So another sheep's egg cell is removed, and the unfertilized egg cell is there. So basically you denucleated this cell, nucleus is removed from the cell, so you have the egg of the sheep, but the nucleus is not there, and you are taking the nucleus of this adult cell and putting it in the this cell. So one sheep gave you egg without nucleus, other sheep gave you a cell with nucleus, but this is an adult cell, so you take the nucleus from this one and enter into the this egg. And now this is done, the cell fusion is done, fertilization will happen, and then the cell division occurs, embryo will be formed, and this embryo is implanted excuse me, I'm sorry about that. The embryo is implanted in the surrogate mother, and then the mother basically give birth to another sheep, and that sheep was identical to this adult, right, and the sheep was named Dolly. Now what is the application of all this? So you are preserving the elite genetics, क्योंकि आप जिस adult से ले रहे हो, that you think is elite one, right? So you are preserving the genetics, and conservation of the endangered breeds are also, this is very important, you do this for conservation also. But there are ethical issues, because you are making a clone, that is not ethically correct, you cannot make a clone, right? It is not permitted also. High cost is also required, low success rate, because Dolly also dies after a few months, right? So health concerns in the clones is always there, because there is premature aging, because this nucleus is not in the early stage of the life, right? You have taken the nucleus from the adult cell, so it behaves like that. So these are some concerns associated with this. Now there is transgenics, which is the emerging field. Now this introduced the foreign genes into the livestock to enhance trade. So again, this is again laboratory based. So basically you are taking the foreign genes and you are introducing it. And how do you introduce the foreign genes? You use a vector, cloning vector. So basically this is your transgene. These are the somatic cells. So transfection mein what you do, this vector will basically infect your cells and introduce the gene. You can see the red color, cells with the integrated transgene is there, and again the transfer of the cell into the cytoplasm is there. Enucleated oocyte mein you are transferring this particular gene, which is your required one, and then again you see that this is basically expressing in your desired your desired gene is going to express, because you culture it and you transfer the embryo in the recipient animal, and this is the delivery of the clone calf, right? So this is how you do it. It may be again the same thing, you are basically taking the oocyte from a mother, and then you are enucleating the oocyte, you are removing the nucleus and entering this one. ठीक है, transfer of a cell into the ooplasm. So again this is kind of similar to what we have done previously. Examples are experimental transgenic cattle resistant to the mastitis. These have been done sheep with altered wool characteristic, even the cattle with improved milk production. We have already done this stages. If you see it, still under the research, regulatory and consumer acceptance issue is there, because people don't want transgenic material. If commercialization is not that good, there are limitations to that. Then there is controlled mating system. So this we have already talked about earlier also. So there is crossbreeding. So the crossbreeding can be of two types: the rotational and terminal crossbreeding. In rotational crossbreeding, the systematic mating of different breeds in rotation is there to maintain the heterosis, which is the hybrid vigor. That means always hybrid will have the enhanced character as compared to the parents, right? Positive characters will increase. In terminal crossbreeding, the F1, the first filial crossbreds, are slaughtered, and the females retained for breeding are crossed with the terminal sires. So F1 crossbreds, you are slaughtering them, and females are retained for breeding, and they are crossed with the terminal sires. What is the advantage? Advantage is that the production will improve, and the trait which you want, using the sires of the desired trait, you increase the production rate, right? Then there is a maintenance of the hybrid vigor across the generation. This is this most important thing of this. So these are the all methods of the modern technology, or the modern methods used for the breeding of the cattle and the sheep. So advantages अगर आप देखेंगे, so rapid genetic improvement is there, improved disease control is there, efficient use of elite sires and dams, and better management of inbreeding, and finally the conservation of the rare breed, very very important, through the gene bank and the embryo preservation. What are the limitations? Especially some technology requires initial cost that is very very high, and this need of the technical expertise. A normal person cannot do it. A trained personnel is also allowed to do it. Ethical constraints are there, and the farmers' awareness and adoption of some technology remains challenged in the developing region of the world. So this is it for today. I hope you understand something. If you have any question or query, feel free to comment it down, and if you understand something, do like this video and subscribe to the channel if you are new. Thank you so much, and I will see you all in the next video lecture.